Picture this. In a lab somewhere in the world right now, there's a cluster of human brain cells about the size of a lentil firing electrical signals. These tiny brain organoids can't think or feel, at least not yet. What if I told you that scientists aren't just growing mini brains that might one day think? They're growing miniature versions of nearly every organ in your body. Hearts that beat, livers that detoxify, intestines that digest, all from a few of your skin cells. Now, I know what you're thinking. Lab-grown organs? Sounds like science fiction. And honestly, 5 years ago, you'd be right. But here's the thing. This isn't some far-off fantasy. Right now, as we speak, researchers are using these miniature organs to test drugs for COVID-19, model Alzheimer's disease, and even figure out why some people's cancer responds to treatment while others don't. Let me paint you a picture of what we're really talking about here. Imagine taking a small sample of your blood or skin. We're talking about something as simple as what happens when you get your blood drawn at the doctor's office. Scientists can take those cells, reprogram them back into stem cells. Think of it as hitting the biological reset button, and then coax them to grow into three-dimensional miniature versions of your organs. We're not talking about full-size replacement organs here. These are typically just a few millimeters across, about the size of a pea. But here's the kicker. They function like the real thing. These organoids are bridging a massive gap in medical research. For decades, we've been stuck between two not-so-great options. Testing on animals, which often doesn't translate well to humans. Ever wonder why that miracle mouse cure never makes it to market? Because mice aren't people. Or testing on flat layers of cells in petri dishes. About as representative of your complex 3D organs as a stick figure is to the Mona Lisa. But organoids, they're the Goldilocks solution. They're human tissue, so no more guessing games about whether what works in mice will work in us. They're three-dimensional and functional, capturing the complex architecture and behavior of real organs. And perhaps most importantly, they can be made from your cells, opening the door to truly personalized medicine. Welcome to Apprentice.io's Industry Insights. I'm Frank Maggiore, and today we're exploring one of the most mind-blowing developments in modern medicine. Organoids, the tiny lab-grown organs that are revolutionizing everything from drug testing to personalized medicine. Think about the implications here. Got a weird reaction to a medication? Doctors could test dozens of alternatives on your own mini organs before you swallow a single pill. Family history of a genetic disease? They can model it in a dish and test potential interventions before you show any signs of the symptoms. Need to know if that expensive cancer treatment will actually work for you? Test it on your own tumor organoids first. But here's where it gets really wild and why this matters to every single person listening. We're not just talking about better drug testing or disease modeling. We're talking about a fundamental shift in how we understand and treat human health. These tiny organs are teaching us about how our bodies develop, how diseases take hold, and how we might fix what goes wrong. Some researchers are even exploring whether these organoids could one day supplement failing organs. Imagine microscopic insulin factories helping diabetics, or mini liver units assisting with detoxification. And yes, before you ask, this does raise some fascinating questions. Human brain organoids are artificially grown, small-scale tissue constructs that resemble the immature brain. And as they become more sophisticated, we're grappling with questions that sound straight out of a philosophy textbook. When does a cluster of brain cells become something more? It's a question scientists and ethicists are actively debating, especially as brain organoids bear an uncanny resemblance to a miniaturized brain and show increasingly complex activity. But let's not get ahead of ourselves. Today, we're going to take you on a journey through the miniature organ revolution. We'll explore how these tiny marvels are made, what they can do right now, and where this technology is headed. We'll meet the different types of organoids, from mini brains solving neurological mysteries to mini guts revealing the secrets of our microbiome. We'll dive into how companies are scaling up from one organoid to millions. And yes, we'll tackle those big ethical questions head on. Most importantly, we'll show you why this isn't just another cool science story. It's a technology that could fundamentally change your healthcare, maybe sooner than you think. Because we're not quite at the point of 3D printing replacement organs, though we'll certainly talk about that in a future episode. We are at the point where your doctor might soon be testing treatments on your own personalized mini organs before they ever enter your body. So buckle up, because we're about to shrink down and explore the big future of these tiny organs. Trust me, by the end of this episode, you'll never look at medical research the same way again. So what exactly are these mysterious organoids that have scientists so excited? Let me break it down for you in a way that won't require a PhD to understand. Imagine you're a baker, but instead of flour and yeast, you're working with human cells. And instead of bread, you're trying to grow a miniature organ. That's essentially what organoid scientists do every day. They take stem cells. These are the Swiss Army knives of the cellular world, capable of becoming any type of cell in your body, and then coax them into forming tiny three-dimensional structures that look and act remarkably like real organs. And here's the wildest part. We're not talking about building these organs cell by cell, like constructing a Lego set. No, these cells actually self-organize. You give them the right environment, the right chemical signals, and they remember what they're supposed to become. It's like watching a pile of Lego blocks spontaneously assemble themselves into a castle. The cells talk to each other, arrange themselves into the proper structures, and before you know it, you've got mini brains firing neurons, or mini guts producing digestive enzymes. The journey from stem cells to organoids is fascinating. Scientists typically start with either pluripotent stem cells or adult stem cells taken directly from specific organs. They place these cells in a special gel. Think of it as a cellular apartment building that gives the cells a place to live and grow into their three-dimensional structures. Then comes the secret sauce, a cocktail of growth factors and signaling molecules that tell the cells what to become. Now, let's clear up some confusion. Organoids are not the same as those flat cell cultures that you might have seen in science class. Traditional cell cultures, they're looking like a map of New York City. Useful, but missing all the skyscrapers and subway systems that make the city actually function. Organoids are more like a scale model of Manhattan, complete with different neighborhoods, infrastructure, and all the complex interactions that make a city work. They're also not organs, at least not yet. A kidney organoid can filter substances and produce urine-like fluids, but it's missing the complex blood vessel networks and full functionality of a real kidney. Think of organoids as organ understudies. They know the lines and can perform many of the same functions, but they're not quite ready for the leading role. Here's where it gets really interesting. Organoids can exist in what I like to call the Goldilocks zone of biological research. They're complex enough to capture the essential features of human organs, the different cell types, the 3D architecture, the basic functions, but simple enough that we can grow them in a lab, manipulate them, and study them in ways we never could with actual human organs. The development process is remarkably similar to how organs form in a developing embryo. The cell sources for establishing organoids include embryonic stem cells, ESCs, adult stem cells, ASCs, induced pluripotent stem cells, iPSCs, and tumor cells. Scientists have essentially hacked into nature's instruction manual, using the same signals that guide organ development in the womb to create these miniature organs in a dish. What makes this possible is our growing understanding of stem cell biology. We've learned that cells don't just follow a predetermined script. They're constantly listening to their environment, responding to chemical signals from their neighbors, and making decisions about what to become. By controlling these signals, we can guide stem cells down to specific developmental paths. The timing is crucial, just like baking, where adding ingredients in the wrong order could ruin your cake. Growing organoids requires precise timing of different growth factors. Add a certain signal too early, and your brain organoid might never develop proper neurons. Add it too late, and you might miss the critical window for development. Size matters too. Current organoids typically max out at a few millimeters, about the size of a lentil. Why so small? Because unlike real organs, organoids don't have blood vessels to deliver oxygen and nutrients to their core. Once they grow beyond a certain size, the cells in the center start to die from lack of oxygen. It's like trying to build a skyscraper without elevators. You can only go so high before it becomes impractical. But even at this tiny scale, the complexity is mind-boggling. A brain organoid the size of a pea can contain millions of cells organized into distinct regions that mirror the structure of a developing brain. Liver organoids produce bile and metabolize drugs. Intestinal organoids form tiny crypts and villi, just like the structures in your gut that help absorb nutrients. The real magic happens at the molecular level. These cells are running the same genetic programs, producing the same proteins, and performing the same functions as cells in your body. When researchers analyze the gene expression patterns in organoids, they find that they're strikingly similar to actual human organs at various stages of development. This brings us to one of the most exciting aspects of organoid technology. They're incredibly personal. Because they can be grown from a patient's own cells, they capture that individual's unique genetics. Your organoids would respond to drugs differently than mine, just like our bodies do. It's personalized medicine at the cellular level. Now that you understand what these tiny organoids are and how they're made, let's meet the all-star lineup. From mini brains to mini guts, each type of organoid has its own superpowers. Alright, let's take a tour through the organoid factory and meet some of the star players. Each type of organoid is like a different superhero with unique powers. And trust me, some of these abilities are straight out of comic books. Let's start with the rock stars of the organoid world, brain organoids. These mini brains are perhaps the most fascinating and controversial of all the organoids. Picture a tiny ball of neural tissue about the size of a pea, containing millions of neurons firing in coordinated patterns. But here's what brain organoids can actually do. They develop distinct regions that mirror different parts of the human brain. They've got areas that look like the cerebral cortex, the hippocampus, and even primitive eye structures in some cases. Researchers are using them to study everything from autism to Alzheimer's disease, watching in real time how these conditions develop at the cellular level. It's like having a window into the human brain that we've never had before. One research team even hooked up brain organoids to robot bodies, creating a bizarre hybrid where lab-grown neural tissue learned to control mechanical systems. We're not talking Terminator level stuff here, but it's still pretty wild to think about neurons grown in a dish learning to interact with the physical world. Next up, liver organoids, the detox champions. Your liver is basically your body's chemical processing plant, and liver organoids are surprisingly good at replicating this function. These mini livers can metabolize drugs, produce bile, and even model diseases like fatty liver disease. Pharmaceutical companies are particularly excited about these because they can now test how new drugs are processed without risking human subjects. Here's a practical example. Say a drug company wants to know if their medication might cause liver damage. Instead of waiting for clinical trials and potentially harming human patients, they can now test it on hundreds of liver organoids first. These tiny organs will process the drug just like a human liver would, showing any signs of toxicity. It's faster, cheaper, safer, and way more ethical than traditional testing methods. Moving down to the digestive system, we have intestinal organoids, your gut feeling in a dish. These are some of the most successful organoids in terms of mimicking their full-size counterparts. They form all the characteristic structures of the intestine, the finger-like villi that absorb nutrients, the crypts where stem cells hide out, even some immune cells that patrol your gut. What's really cool is that intestinal organoids can be infected with actual pathogens. Researchers have used them to study how norovirus, that nasty stomach bug that sweeps through cruise ships, infects human cells. They've watched in real time as bacteria like Salmonella invade the intestinal walls. It's like having a microscopic battlefield where we can study the eternal war between our bodies and disease-causing microbes. But wait, there's more. Heart organoids that actually beat. Scientists have created tiny heart chambers that contract rhythmically, pumping fluid just like your heart pumps blood. They're using these beating mini hearts to study genetic heart conditions, test cardiac drugs, and understand how heart attacks damage tissue. Imagine being able to give a patient with a genetic heart condition a drug that's been tested on their own cardiac tissue. That's the promise of heart organoids. Lung organoids are helping us understand respiratory diseases in ways that we never could before. During the COVID-19 pandemic, researchers used lung organoids to study how the virus infects airway cells and causes damage. They can watch the infection process in real time, test potential treatments, and understand why some people get sicker than others. These mini lungs have air sacs, produce mucus, and even have some of the cellular defenses that protect our airways. Kidney organoids are perhaps the most structurally complex, forming tiny filtering units called nephrons. Implantation of pancreatic islet-like organoids generated by planting pancreatic islet cells into a bioscaffold has the ability to restore insulin secretion and decrease glucose levels in type 1 diabetic mice. While we're not quite at the point of using organoids as replacement organs, these studies show the incredible potential. Now, let's have that reality check I promised. Current organoids are like talented actors who know their lines but haven't quite mastered their full performance. Brain organoids can fire neurons but can't think. Liver organoids can detoxify but can't produce all the proteins a full liver can make. Heart organoids can beat but can't pump blood through a body. The biggest limitation? Vascularization. The blood vessel problem I mentioned earlier. Real organs have intricate networks of blood vessels delivering oxygen and nutrients to every cell. Organoids don't, which limits their size and complexity. It's like trying to run New York City without a subway or a road system. Things break down pretty quickly. There's also the maturity issue. Most organoids resemble fetal or infant tissue rather than adult organs. The iPSC-derived organoids present low maturation, which are similar to embryonic or early fetal tissues, and can be used in studying organogenesis. This is great for studying development, but limits their use in modeling adult diseases. And then there's the variability problem. Even when following the exact same protocol, two organoids might develop differently. It's like baking sourdough. Even with the same recipe, every loaf comes out a little different. This biological variability is natural, but makes standardization challenging. Despite these limitations, the achievements are staggering. We've gone from growing simple cell layers to creating complex, functional mini organs in just over a decade. Each type of organoid opens new doors for understanding human biology and disease. Okay, so we've got these tiny organs growing in labs around the world. Very cool, very sci-fi. But here's the multi-billion-dollar question. What can we actually do with them that matters to real people with real health problems? Turns out, quite a lot. Let's start with disease modeling, or creating a disease in a dish. This is where organoids really shine. Imagine you're trying to understand Alzheimer's disease. You could study mice, which don't naturally get Alzheimer's. You could wait for donated brain tissue from deceased patients, by which point the disease has already done its damage. Or you could grow brain organoids with Alzheimer's causing mutations and watch the disease unfold in real time. And that's exactly what researchers are doing. They're taking cells from Alzheimer's patients, growing them into brain organoids, and watching as protein tangles form, neurons die, and inflammation spreads. It's like having a time-lapse video of a disease that normally takes decades to develop. For the first time, we could see the very earliest stages of these conditions, the molecular dominoes that start falling years before the symptoms appear. Cystic fibrosis is a genetic disease that affects different people in dramatically different ways. Using organoids, doctors can now grow mini lungs or mini intestines from specific patient cells and test which drugs work best for their exact genetic mutation. This isn't theoretical, it's happening right now. Kids with cystic fibrosis are getting personalized treatment plans based on how their own organoids respond to different medications. Moving on to drug discovery, this is where organoids could save the industry literally billions of dollars and countless lives. Here's a sobering statistic: about 90% of drugs that enter clinical trials fail. Why? Often because what works in mice doesn't work in humans, or simply the underlying mechanisms work differently in humans. But organoids can bridge that gap. Take cancer research. Organoids can be used as disease models to understand the mechanisms and physiopathology of human diseases. Organoids are ideal models for drug screening and toxicity assays. Patient-derived organoids can be used to predict patient-specific responses to drugs and personalized treatments. Instead of treating all liver cancers the same way, doctors can now grow organoids from a patient's tumor and test dozens of drug combinations to find the most effective treatment. It's like having a practice run before the real game. Pharmaceutical companies are all in on this technology. They are using organoids to screen thousands of potential drug compounds quickly and cheaply. The object here is if you're going to fail, you fail fast and you fail cheaply. A process that used to take years and cost millions of dollars can now be done in weeks for a fraction of the price. And because they're testing on human tissue, the results are far more relevant than animal studies. But perhaps the most exciting application is personalized medicine. The idea that medical treatment should be tailored to you specifically, not just your disease. This is where organoids truly transform from cool science to life-changing technology. Here's a real-world scenario. A patient has colorectal cancer. Their doctors aren't sure which chemotherapy cocktail will work best for that patient. In the old days, they'd try one, wait to see if it works, maybe try another if it doesn't. Months of trial and error, with the patient suffering severe side effects from drugs that might not even help. Enter organoid technology. Doctors take a small biopsy of the patient's tumor, grow organoids from it, and test different drug combinations. Within weeks, they know that drug A kills 80% of their cancer cells. Drug B only kills 30%, and drug C actually makes them grow faster. The patient gets drug A, it spares them from the worst side effects, from ineffective treatments, and the patient has a much better chance of beating their cancer. This isn't science fiction, it's happening in cancer centers around the world. The Dutch have been pioneers here, creating living biobanks of patient organoids. It's like a library of mini tumors that researchers can use to develop and test new treatments. The organoid biobank concept is revolutionary. Imagine having a library of mini organs representing different diseases, genetic backgrounds, and patient populations. Researchers can literally check out organoids like library books, test their hypotheses, and return them for others to use. We're building a renewable resource for medical research that doesn't require constant patient participation. These biobanks are particularly valuable for rare diseases. If you have a condition that affects only a few thousand people worldwide, drug companies historically haven't invested in finding treatments. The market is just too small. But with organoid biobanks, researchers can study these rare conditions without needing access to patients, making drug development easier and more feasible. The applications extend beyond just testing existing drugs. Organoids are helping us to understand why certain people respond to treatment while others don't. They're revealing new drug targets, suggesting novel combination therapies, and even helping predict side effects before a drug ever enters a human body. Environmental toxicology is another huge application. We're surrounded by thousands of chemicals in our food, our cosmetics, and cleaning products. Which ones might cause cancer, birth defects, organ damage? Instead of testing them on animals or waiting for epidemiological evidence, by which point people have already been harmed, we can test these chemicals on human organoids. The COVID-19 pandemic showcased organoids' potential in responding to emerging infectious diseases. Within weeks of the virus emerging, researchers worldwide were infecting lung and intestinal organoids to understand how the virus attacks human tissues. This research directly informed treatment strategies and helped explain why some people got so much sicker than others. So, we can grow them, we can use them for research, but here's the trillion-dollar question: how do we scale this up? How do we go from one precious organoid in a university lab to millions of standardized mini organs that can transform medicine? Time to talk about the manufacturing challenges. Alright, let's get real about the elephant in the room. Growing one organoid in a lab? That's biology. Growing millions of identical organoids with consistent quality? That's engineering. And folks, this is where things get seriously complicated. Think about it this way. Imagine you perfected the world's best sourdough recipe. You can make one perfect loaf in your kitchen. Now someone asked you to make 1,000,000 loaves, all exactly the same, every single day, in facilities around the world. Oh, and even if one loaf is slightly off, someone could die. Welcome to the world of organoid manufacturing. The challenges are staggering. First, there's the consistency problem. Living cells are not machines. They're more like temperamental artists. Each batch of stem cells might behave slightly differently. The growth factors you add might vary in potency. The temperature fluctuates by half a degree, and suddenly your brain organoids are developing differently. When you're making drugs that people's lives depend on, close enough just doesn't cut it. Here's where 3D bioprinting enters the picture, and it's as cool as it sounds. Bioprinting provides a precise and controlled environment for organoid placement and growth, which enables the production of highly organized and reproducible 3D tissue structures. Instead of letting organoids self-assemble randomly, bioprinters can precisely position cells in three-dimensional space, almost like an incredibly sophisticated 3D printer, but printing with living cells instead of with plastic. These aren't your desktop 3D printers either. We're talking about machines that can deposit individual cells with micrometer precision, layer by layer, building structures that mimic human tissue architecture. Some use inkjet-like technology, spraying tiny droplets of cells. Others use extrusion, squeezing out cell-laden gels like the world's most precise pastry bag. The newest techniques use light to solidify cell-containing gels in complex 3D patterns, building entire structures in seconds rather than hours. But here's the challenge. Cells are fragile. Too much pressure and they die. Too little and they won't stay where you put them. The bio-ink, the material carrying the cells, needs to be liquid enough to flow through tiny nozzles, but solid enough to hold its shape once it's printed. It's a material science nightmare that's making regular 3D printing look like child's play. Quality control is another massive hurdle. How do you check if an organoid is good without actually destroying it? You can't just take a sample like you would with a chemical drug. Advanced imaging systems are being developed that can peer inside organoids without needing to damage them, using techniques like optical coherence tomography, basically ultrasound, but with light, to verify their structure. Now this is where cloud-based manufacturing execution systems become absolutely critical. We're not just talking about tracking widgets in a factory, we're monitoring living systems that change over time. Cloud-based manufacturing execution systems are particularly valuable for organoid manufacturing because they can handle the incredible complexity of managing biological processes. Every parameter needs to be tracked temperature, pH, oxygen levels, growth factor concentrations, even the batch number of the plastic dishes you're growing them in. These manufacturing execution system platforms, like Apprentice.io's Tempo Manufacturing Cloud, are the nervous system of modern biomanufacturing. They're tracking thousands of variables in real time, using AI to predict when batches might fail, and ensuring that every single organoid can be traced back to its source. If there's a problem with organoids delivered to a hospital in Tokyo, manufacturers need to instantly know which batches might be affected, what raw materials were used, and what other customers might have received similar products. The data management alone is mind-boggling. The manufacturing execution systems needs to not just store this data, but to make it instantly searchable and analyzable. Machine learning algorithms continuously analyze this data, looking for patterns that humans might miss. Let's talk about the economics for a moment. Current estimates suggest that producing therapeutic-grade organoids can cost hundreds or even thousands of dollars per unit. That's fine for research, but if we want to use organoids therapeutically, say as mini insulin factories for diabetes, we need to drive those costs way down. This means optimizing every step of the process, from the cost of growth media to the efficiency of bioreactors. Scale-up strategies are evolving rapidly. Some companies are developing bioreactor systems that can grow multiple organoids simultaneously under carefully controlled conditions. Others are working on microfluidic systems, tiny chips with channels smaller than a human hair, where individual organoids can be grown in isolated chambers but maintained collectively. The regulatory landscape adds another layer of complexity. Organoids fall into a regulatory gray area. They're not quite drugs, not quite medical devices, and not quite transplant organs. Regulatory agencies worldwide are scrambling to develop frameworks for ensuring their safety and efficacy. Every aspect of the manufacturing process needs to be documented, validated, and approved. But here's the exciting part. We're actually solving these problems. The 3D bioprinting of organoids is still in its infancy. The technology holds promise for the engineering of multiple organoid tissue constructs. Companies are building GMP-certified facilities specifically designed for organoid production. Automation systems are becoming more sophisticated. Companies are building facilities specifically designed for organoid production. Costs are dropping as processes are optimized. The integration of different technologies is key. It's not just about having good bioprinters or sophisticated bioreactors or even powerful manufacturing execution systems. It's about making them all work together seamlessly. When a bioreactor creates organoids, the manufacturing execution system needs to immediately assign them unique identifiers, track their location, and begin monitoring their development. When those organoids are ready for quality control, automated imaging systems need to access them and feed that data back into the MES. This manufacturing revolution isn't happening in isolation. It's part of a broader transformation in how we produce biological medicines. The same technologies enabling organoid manufacturing are also advancing cell therapies, gene therapies, and other cutting-edge treatments. We're building the infrastructure for a new era of medicine. Now that we've covered how to mass produce these tiny organs, let's explore the cutting edge, the wild, wonderful, and sometimes weird ways researchers are pushing organoid technology to its limits. If you thought growing mini organs was crazy, wait until you hear what scientists are doing with them now. Let's start with the brain-computer interface work that's making headlines. Neurons were successfully embedded in digital game worlds. The biologically inspired neural network, expressing human cortical cells, was able to show internal modification and learn the task at hand, predicting the trajectory of a digital ball while moving a digital paddle. That's right, scientists taught lab-grown brain tissue to play Pong. Not metaphorically, but literally. They connected brain organoids to computer systems, fed them information about a digital ball's position, and the neurons learned to control a digital paddle to hit them. This isn't just a party trick. This research is revealing fundamental insights about how neural networks learn to adapt. The brain organoids showed that they could process information, learn from feedback, and improve their performance over time. It's raising profound questions about consciousness, intelligence, and what it means to be a thinking system. Questions about consciousness and brain organoids are becoming urgent as the science continues to develop. And they're questions we need to answer before this technology goes much further. Now let's talk about organoid-on-a-chip technology, which is exactly as futuristic as it sounds. Scientists are creating microfluidic devices. Think of them as tiny plumbing systems carved into chips the size of a credit card, where different organoids can be connected and communicate. You might have a liver organoid in one chamber, a heart organoid in another, and a kidney organoid in a third, all connected by tiny channels that allow them to share chemical signals. And why does this matter? Because in your body, your organs don't work in isolation. Your liver processes drugs, creating metabolites that might affect your heart or your kidneys. Traditional drug testing looks at these organs individually, missing these crucial interactions. But with organ-on-a-chip systems, we can model how drugs affect multiple organs simultaneously. It's like having a tiny human on a chip, minus the consciousness and the ethical concerns. One team created a body on a chip with 10 different organ types, including brain, liver, kidney, and skin organoids. They used it to test cancer drugs, watching in real time how the drug was metabolized by the liver, how those metabolites affected the heart, and whether they crossed the blood-brain barrier. Speaking of breakthrough applications, let's talk about transplantable organoids. We're not quite at the point of growing replacement organs, but we're taking the first steps. Implantation of pancreatic islet-like organoids, generated by planting pancreatic islet cells into a bioscaffold, has the ability to restore insulin secretion and decrease glucose levels in type 1 diabetic mice. While these are still the early days, the implications are staggering. Imagine a future where type 1 diabetics don't need to get insulin injections because they have organoid insulin factories implanted under their skin. Or where patients with liver failure can receive liver organoid transplants to boost their organ function while they're waiting for a full transplant. We're not there yet, but the path is becoming clearer. The race for functional mini organs is intensifying. Recent breakthroughs include liver organoids that can partially restore liver function in mice with liver failure, tear gland organoids that can actually cry, produce tears when stimulated, mammary gland organoids that produce milk proteins, tooth bud organoids that could potentially grow new teeth. But perhaps the most mind-bending development is in the field of assembloids, combination of different organoid types that model organ interactions. Scientists have fused brain organoids with spinal cord organoids and watched as the neural connections form between them. They've connected brain organoids with muscle tissue and observed the brain tissue learning how to control those muscle contractions. One particularly striking example, researchers created an assembloid combining human brain organoids with eye organoids. The eye structures develop light-sensitive cells, and when exposed to light, they send signals to the connected brain tissue. We're literally watching the development of primitive visual systems in a dish. The vascularization problem I mentioned earlier, scientists are attacking it from multiple angles. Some are 3D printing blood vessel networks within organoids. Others are coaxing organoids to grow their own blood vessels by adding endothelial cells and the right growth factors. One team even developed bloodless blood vessels, channels that deliver nutrients without actual blood cells, solving the oxygenation problem while avoiding the complexity of a full circulatory system. Here's where artificial intelligence enters the picture in a big way. AI systems are now designing optimal protocols for growing organoids, predicting which combinations of growth factors will produce the best results. Machine learning algorithms analyze thousands of organoid images, identifying subtle features that predict whether an organoid will develop properly or fail. It's accelerating research by orders of magnitude. The intersection of organoids and gene editing is producing remarkable results. Using CRISPR technology, scientists can introduce specific genetic mutations into organoids to model diseases. But they're going even further. They're using gene editing to correct genetic defects in patient-derived organoids, demonstrating that these diseases could potentially be cured at the cellular level. One team took organoids from patients with cystic fibrosis, using CRISPR to correct the genetic mutation, and showed that the fixed organoids function normally. It's a proof of concept that combines organoid technology with gene therapy, potentially offering cures for genetic diseases that were previously untreatable. The applications are getting even more creative by the day. Cosmetic companies are using skin organoids to test products without needing to use animal testing. Food scientists are using taste bud organoids to develop new flavors. Environmental scientists are using lung organoids to study air pollution effects. The technology is spreading far beyond medical applications. But here's what really excites me about the cutting edge. We're seeing convergence. Organoids aren't developing in isolation. They're being combined with robotics, AI, gene editing, nanotechnology, and other advancing fields. Future research in engineering structured organoids will likely be shaped by three key approaches: 3D bioprinting, organoid-on-chip systems, and artificial intelligence. Each combination opens up new possibilities we couldn't have imagined just a few years ago. Now I know what some of you are thinking. This all sounds amazing, but also kind of concerning. Growing brains that can learn, creating human tissue at scale. Time for that reality check about challenges and ethical considerations that we need to grapple with before we end up in a Black Mirror episode. Okay, time to pump the brakes a bit and talk about the real challenges and some pretty heavy ethical questions. Because let's be honest, when you're growing human brain tissue that shows electrical activity, you've entered territory that would make Mary Shelley nervous. Let's start with the technical hurdles that are keeping organoids from reaching their full potential. The vascularization problem remains the biggest roadblock. Without blood vessels, organoids hit a size ceiling. It's like trying to build a city without roads. You can only get so big before transportation becomes impossible. Scientists are making progress, but we're still years away from organoids with fully functional circulatory systems. Then there's the maturation issue. Most organoids are stuck in a perpetual state of youth, resembling fetal or infant tissue. This is great for studying development, but not so great for modeling adult diseases. It's like trying to study retirement planning with a classroom full of kindergartners. You're missing some crucial perspectives here. The complexity gap is real too. Your liver has over 500 functions. Current liver organoids can handle maybe a dozen of those. Brain organoids might have 6 types of cells, while real brains have hundreds. We're playing a symphony with a fraction of the orchestra. Standardization remains elusive as well. Two labs following identical protocols might get very different results. It's like following the same recipe, but getting chocolate chip cookies in one kitchen and snickerdoodles in another. This variability makes it hard to compare research results or develop standardized therapeutic products. But now let's dive into the really thorny stuff, the ethical considerations. And friends, this is where things get philosophically intense. The consciousness question is the elephant in the room when it comes to brain organoids. All this suggests that in the future, human cerebral organoids may manifest an ability to experience basic sensations such as pain, therefore manifesting sentience, or even rudimentary forms of consciousness. This isn't just philosophical navel-gazing. If brain organoids can feel pain or have experiences, even rudimentary ones, then do we have moral obligations towards them? But how do you determine if a blob of brain tissue is conscious? We don't even fully understand consciousness in humans, let alone in an isolated lentil-sized mass of neural tissue. Some ethicists have proposed the precautionary principle. If we can't be sure organoids aren't conscious, then we should err on the side of caution and establish ethical guidelines for their treatment. Others argue we're nowhere near the complexity needed for consciousness. But as brain organoids become more sophisticated, showing more complex activity patterns, this debate will only intensify. The regulatory landscape is a maze. Organoids don't fit neatly into existing categories. Are they human tissue, medical devices, drug products? Different countries are approaching this differently, creating a patchwork of regulations that make international research challenging. Companies developing organoid-based therapies face regulatory uncertainty that can delay or derail promising treatments. There are also concerns about commercialization and consent. If someone donates cells for organoid research, who owns the resulting organoids? Can they be patented? Sold? What if those organoids lead to a blockbuster drug? Does the cell donor get a cut? These aren't hypothetical questions. They're being debated in boardrooms and courtrooms right now. The humanization issue is particularly touchy when it comes to transplanting human organoids into animals for research. If you put human brain organoids into a mouse brain and those organoids integrate and function, have you created something that's neither fully mouse nor fully human? Where do we draw the ethical lines? Then there's the question of enhancement versus treatment. If we can grow better organoids, say liver organoids that are more efficient than natural livers, should we use them to enhance healthy people, such as in detoxifying someone's blood after a night of binge drinking? Or should this technology be restricted to treating diseases only? It's the same debate we're having about gene editing, but with an organoid twist. Safety concerns are paramount. We're still learning about the long-term behavior of organoids. Could transplanted organoids become cancerous? Might they develop in unexpected ways? The preclinical testing required to ensure safety is extensive and expensive, potentially limiting access to these treatments. The path from lab curiosity to FDA approval is long and winding. A typical drug takes 10 to 15 years to go from discovery to pharmacy shelves. Organoid-based therapies might take even longer, given the regulatory uncertainty and the need to establish entirely new safety and efficacy standards. Public perception is another challenge. While scientists see organoids as powerful research tools, the public might see lab-grown brains and think of science fiction horror stories. Clear, honest communication about what organoids are and aren't is crucial for maintaining public trust and support. But here's the thing, none of these challenges are insurmountable. The technical hurdles, scientists are chipping away at them every day. The ethical questions, bioethicists, researchers, and policymakers are working together to develop frameworks that promote beneficial research while protecting against misuse. The regulatory uncertainty? Agencies worldwide are developing new guidelines specifically for organoid technologies. What we need is thoughtful, inclusive dialogue about how to develop this technology responsibly. We need diverse voices, not just scientists and ethicists, but patients, caregivers, and communities who might benefit from or be affected by organoid technology. We need transparency about both the promise and the limitations. All right, we've covered a lot of ground today, from the science to the challenges, from the tiny beating hearts to big ethical questions. But what does this all mean for you, for medicine, for our future? As we wrap up our journey through this miniature organ universe, we've seen brain organoids learning to play video games, mini hearts beating in dishes, and tiny intestines fighting off infections. We've grappled with consciousness in a petri dish and manufacturing challenges that would make rocket science look simple. But what does it all mean? Where is this heading? And why should you care? Let me paint you a picture of the future that's not far off, about the next decade in the future for organoids. Imagine walking into your doctor's office with a mysterious illness. Instead of the current trial and error approach of treatment, your doctor takes a small sample of your cells. Within weeks, a lab has grown organoids from your tissue and tested dozens of treatments to find the one that works specifically for you. No more being a guinea pig for medications that might not work, or even worse, might harm you even more. No more suffering through side effects from the wrong drugs. Or picture this, a child is born with a rare genetic disease that may affect maybe 100 people worldwide. In the past, that child would have faced a lifetime of symptoms with no hope of treatment. The disease is too rare for pharmaceutical companies to invest into it. But with organoid technology, researchers can model that exact disease, test thousands of potential treatments, and develop a therapy tailored specifically to that condition. We focus on the applications of organoids in basic biology and clinical research, and also highlight their limitations and future perspectives. The convergence we're seeing is extraordinary. AI is designing better ways to grow organoids. Robotics is automating their production. Gene editing is allowing us to correct diseases at the cellular level. Bioprinting is giving us precise control over their structure. Each advance amplifies the others, creating exponential progress. Within the next 5 to 10 years, I predict we'll see organoid-based drug testing becoming standard in pharmaceutical development. Personalized cancer treatments based on patient-specific tumor organoids. The first successful organoid transplants for organ support. Brain organoids revolutionizing our understanding of difficult-to-treat neurological diseases. Environmental and cosmetic testing shifting entirely to organoid-based methods. But here's what gets me really excited, the democratization of this technology. As the costs come down and methods standardize, organoid technology won't be limited to major research centers. Smaller hospitals, universities in developing countries, and even high school science classes might have access to basic organoid technologies. Imagine students learning about human biology, not from textbooks, but from watching actual human tissue develop and function. The message of hope is this: we're not just growing organs, we're growing possibilities. Every organoid represents a chance to understand disease better, to develop more effective treatments, to reduce suffering. Every advance brings us closer to truly personalized medicine, where treatments are tailored not just to your disease, but to your unique biology. For patients and families dealing with currently untreatable conditions, organoids offer something precious, hope grounded in science. Not false promises or miracle cures, but real, measurable progress toward understanding and treating diseases that have long seemed insurmountable. But this future isn't inevitable. It requires all of us to play a part. So here's my call to action. For patients and families, ask your doctors about organoid research related to your condition. Many clinical trials are recruiting participants. Your involvement could help accelerate breakthroughs not just for you, but for everyone with your condition. For healthcare professionals, stay informed about organoid technology. This isn't just for researchers, it's rapidly becoming relevant to clinical practice. Push for your institutions to explore organoid-based testing and treatment options. For students and young scientists, this field is wide open for innovation. We need bright young minds from diverse backgrounds, not just biology, but engineering, computer science, ethics, and more. The biggest breakthroughs often come from interdisciplinary thinking. For policymakers and regulators, we need thoughtful, adaptive regulations that promote innovation while ensuring safety. Engage with the scientific community to understand this technology and create the frameworks that enable responsible development. For everyone else, support funding for basic research, share accurate information about organoid technology, participate in public discussions about the ethical implications. Your voice matters in shaping how this technology will ultimately develop. to companies developing manufacturing systems like Apprentice.io with their Tempo Manufacturing Cloud. These are the unsung heroes of this revolution. They're building the infrastructure that will transform organoids from laboratory curiosities to accessible medical tools. Supporting and working with these companies accelerates the entire field. Remember, every transformative medical advance started with skepticism. Antibiotics, vaccines, organ transplants, all were considered impossible or too dangerous. Organoid technology faces similar doubts, but the science is solid, the progress is real, and the potential is extraordinary. We're standing at a unique moment in medical history. We have the knowledge to grow human tissue in labs. We have the technology to study disease in unprecedented detail. We have the tools to develop truly personalized treatments. What we need now is the collective will to push forward responsibly and inclusively. The organoid revolution isn't coming, it's already here, and it needs all of us. Every scientist pushing boundaries, every patient sharing their cells, every policymaker writing thoughtful regulations, every citizen supporting the research. We're all part of the story. We're not just growing organs, we're growing hope. We're growing a future where disease is understood better, where treatment is personal, where rare doesn't mean forgotten, where suffering has solutions, and that future, it's growing in a dish right now, waiting for us to nurture it into reality. Thank you for joining me on this incredible journey into the world of organoids. If this moved you, inspired you, or opened your eyes, share it, talk about it, act on it, because the future of medicine isn't just in the hands of scientists, it's in all of our hands. Until next time, keep questioning, keep hoping, and remember, sometimes the biggest revolutions come in the smallest packages. In an industry where precision and compliance are paramount, Apprentice's Tempo platform offers a cutting-edge manufacturing execution system solution. It's cloud-based, mobile-first, operator-centric, with the most intuitive UI in the industry, making it perfect for today's dynamic pharmaceutical manufacturing environments and for the production of the world's most complex drug products. Whether you're working on groundbreaking cell and gene therapies, biologics, or small molecules, Tempo provides a fully validated MES system that's both powerful and easy to use. It streamlines your processes, ensures compliance, and adapts to your specific manufacturing needs. So get off of paper batch records and discover how Apprentice's Tempo Manufacturing Cloud can transform your operations by visiting us at apprentice.io. Attention industry professionals, revolutionize your manufacturing process with the Apprentice Tempo Manufacturing Cloud, a manufacturing execution system built to streamline your operations, boost efficiency, and ensure compliance all in one powerful platform. Experience the future of manufacturing with our free 30-day trial. Scan the QR code on your screen now to unlock a world of innovation. Don't just keep pace, set the tempo as an industry leader with Apprentice. That's all for Apprentice.io's Industry Insights. I'm Frank Maggiore. Thanks for listening. Be sure to like and subscribe. Hit the bell icon for notifications when new episodes drop. Please comment on if you have any additional topics you would like for me to cover in this space, and we'll see you next week for more Industry Insights.